Hi, this is Lucas Perez, and I did this presentation with my partner Bianca Sanchez. And this is the lab report 7 and 8 combined, the biochemical tests of oxidase test, catalyze test, nitrate reductase test, urea hydrolysis test, CAM test, coagulase test, and Kirby-Bauer susceptibility test. For the introduction, biochemical tests are essential in microbiology for identifying and characterizing bacteria species based on their enzymatic activities and metabolic properties. This report examines the oxidase test, catalyze test, nitrate reductase test, urea hydrolysis test, CAM test, coagulase test, and Kirby-Bauer susceptibility test. Each test reveals distinct bacteria capabilities such as substrate, breakdown, and antibiotic resistance, allowing us to better understand the physiological and pathogenic characteristics of the isolates.
Materials. For the oxidase test, we use the oxidase reagent, a sterile swab or wooden stick, and bacterial culture. For the catalase test, we use the hydrogen peroxide, 3% hydrogen peroxide, a glass slide, and bacterial colony. For the nitrate reductase test, we use nitrate broth with Durham tube, sulfonilic acid, naphthylamine, zinc dust, bacterial cultures. For the urea hydrolysis test, we use the urea agar slants and bacterial cultures. For our CAMP test, we use blood agar plates, Staphylococcus aureus, and Streptococcus galactiae. For the coagulase test, we use coagulase plasma from either rabbit or human and the bacterial culture. For the Kirby-Bauer susceptibility test, we use Moeller-Hinton agar plates, antibiotic discs, and bacterial cultures. The bacterial cultures we used included E. Ruseopathiae, E. coli, Proteus vulgaris, P. aeruginosa, S. aureus, S. epidermidis, S. saprophyticus, S. pyogenes, and S. agalactiae. Methods. So for our oxidase test, we used a sterile swab or a wooden stick to pick up a colony of the bacterial culture. We then applied the colony to a clean glass slide. We add a drop of oxidase reagent onto the colony, and we observed for a color change. Within 30 seconds, a purple color indicated a positive result. For our catalase test, we placed the colony from the agar onto a clean glass slide. We added a few drops of the 3% hydrogen peroxide to the colony. We then observed for bubbling, so the presence of bubbling indicates a positive catalase reaction. For the nitrate reductase test, we inoculated a nitrate broth tube with the bacterial culture and included a Durham tube to detect gas production. We incubated the tube at 35 to 37 degrees Celsius for about 24 to 48 hours. After incubation, we added one dropper of sulfanic acid followed by one dropper of naphthylamine to the broth. If there was no color change, we added a small amount of zinc dust to the tube and we observed for a color change. So a red color indicated a positive test for the nitrate reduction. For the urea hydrolysis test, we streaked a urea agar slant with the bacterial culture. We incubated this slant at 35 to 37 degrees Celsius for 24 hours and we observed it for color changes, with pink indicating urease activity. For our CAMP test, we streaked a blood agar plate with Staphylococcus aureus. We streaked a line of Streptococcus agalactiae perpendicular to the Staphylococcus streak. We incubated this plate at 35 to 37 degrees Celsius for 24 hours. We examined for an arrowhead zone of hemolysis indicating a positive reaction. For our coagulase test, we added a small amount of the bacterial culture to a tube containing coagulase plasma. We incubated it at again 35 to 37 degrees Celsius for 24 hours. And we observed for a clot formation, so the presence of a clot indicates a positive test. Lastly, for our Kirby-Bauer susceptibility test, we prepared a Moeller-Hinton agar plate and inoculated it with the bacterial culture to create a uniform lawn of growth. We allowed the agar to dry for a few minutes After it dried, we placed antibiotic discs. The three that we put were bacitracin, novobiocin, and optochin onto the agar surface. We incubated the plates at 35 to 37 degrees Celsius for 18 to 24 hours, and then we measured the zones of inhibition surrounding each plate.
So for the oxidase test, we found that E. coli and unknown 204 didn't show any color change, which means that they're both oxidase negative. But we have, fortunately, unknown 104 show a full purple color, indicating that it's a strong oxidase test. But we're also kind of like in the middle about P. aeruginosa because we don't know if it show a little bit of oxidase activity or if that's just part of the test. You could see like a little like dot, like black dot on the top of the Q-tip. So we don't know about that one. It probably could be dust. It probably could be some oxidase.
These are our results for the catalase test. So for our S pyogenes, which is on the left, we had a negative result. This was because when we added the hydrogen, it did not produce bubbles, which indicated an absence of the catalase enzyme. And then on the right, we have S aureus, which gave us a positive. So you can see bubbles were produced after hydrogen peroxide was added due to the release of oxygen gas.
For the nitrate reductase test, we have unknown 104, Pierre Reganosa, and E. coli showing all a bright red color, confirming that they can reduce nitrate to nitrite. Unknown 204 show a dark red color, which might suggest doing some partial reduction. But also we have A. vesicalis, which was the exception, showing a transparent color with some floating bacteria around it, suggesting that it didn't manage to reduce nitrate at all. For the CAM test results, we could see good growth in all three lines of the Streptococcus agalactiae, confirming that it's positive for the CAM reaction. This means it's likely enhancing the hemolysis of other bacteria. However, uh, espiogenes show minimal growth in two lines and no growth in the third line.
These are our coagulase results. So for our S. epidermidis, we got a negative test result. It did not produce coagulase, so there was no clotting shown. And that's on the left. And then on the right, we have S. aureus, which gave us a positive test. And you can see the clotting of the plasma, which is like a gel like consistency at the bottom of the test tube. These were our results for the Kirby Bauer susceptibility test. So, unfortunately, because the antibiotic discs fell off the media, we weren't able to interpret the results for the first one, which is P. aeruginosa, the second one, which is E. coli, and the third one, we were only able to measure an inhibition zone for novobiocin on the S. aureus, and it was about a little over half an inch.
So for the first part of the conclusion, we have the oxidase test, the nitrate reductase test, the CAM test, and the Kirby-Bauer susceptibility test. For the oxidase test, we can see E. coli and unknown 204 showing no color change, indicating that they lack the cytochromere C oxidase enzyme. In contrast, P. aeruginosa show a slightly blackish color, suggesting weak oxidase activity, while unknown 104 demonstrated a strong positive reaction with a full purple color, indicating robust oxidase present. As I mentioned, on P. aeruginosa, we're still doubting about that result. For the nitrate reductase test, we can see that unknown 104, P. aeruginosa, and E. coli produce bright red colors, confirming their ability to reduce nitrate to nitrite. Unknown 204 show it a dark red, which may indicate a different reduction pathway or concentration of the nitrite. Conversely, A. fascicalis display a transparent color with floating bacteria, suggesting it did not reduce nitrate effectively. CAM test results indicate significant growth in the three lines of S. agalactiae, confirming its presence and hemolytic activity. There was minimal growth in two lines of S. pyogenes, while the third line showed no growth, suggesting variability in the hemolytic activity of this species. Lastly, the Kirby-Bauer suggestibility test couldn't be interpreted due to the antibiotic disc fouling of the media, which prevented any inhibition of the antibiotics to the bacteria, which ended up leaving us with no inhibition zones, which unfortunately were not able to be examined.
Conclusion. The catalase test helps differentiate catalase positive from catalase negative bacteria by detecting the presence of a catalase enzyme which breaks down hydrogen peroxide into water and oxygen. The urea hydrolysis tests the ability of bacteria to hydrolyze urea into ammonia and carbon dioxide is assessed by the enzyme urease. This is commonly detected using a medium containing urea and a pH indicator. In a positive test, the hydrolysis of urea produces ammonia, which raises the pH and changes the indicator color to pink. A negative test, there is no color change, which means that the organism did not produce urease or it does not hydrolyze urea effectively. The coagulase test is used to differentiate Staphylococcus species based on the production of the enzyme coagulase, which can clot plasma. For our Kirby-Bauer test, this is also known as the disc diffusion method. It is a standardized test used to determine the antimicrobial susceptibility of bacteria. It is widely used in microbiology labs to assess how effective different antibiotics are against the specific bacterial isolate. The results are determined by measuring the zone of inhibition, which is the clear area around the antibiotic disc where where microbacterial growth is inhibited.